A very good morning to all. So we are extremely pleased to have our director, faculty members, and my dear students. I, Dr. Satish Kumar Mittapalli, extend to all a warm welcome to the expert talk on progress of innovation, development, and technology readiness level organized by Amity Institute of Pharmacy, Amity University, Madhya Pradesh, Gwalior, in collaboration with Institutions Innovation Council. Now, I would like to call the Director of Amity Institute of Pharmacy, Dr. S. Mohan Lakshmi Ma'am, to deliver welcome address for this event.
Good morning to all of you. I welcome you all for this expert talk on innovation and technology readiness. So the word innovation, invention, critical thinking, startup, entrepreneurship, these kind of words we are hearing in recent days only. So when we were studying B Pharmacy or when we were in our school days or in our college days, we haven't heard much about these words. These words has become very fancy and buzzword especially in the business world of the present situation. So I was thinking for many days, what is the difference between these words? What is the difference between invention? What is innovation? Is there really any difference, or all these are the same words with the same different words with the same meaning? So when I just started analyzing during the COVID time only, especially these kind of words has become very famous and very talked about words. So when I just personally started analyzing this, so what is this invention? I just want to share with you about these words. So what is the invention and innovation? Because today we are going to talk about we are going to listen about this innovation, and our collaborating body is also Institute Innovation Council. So invention is the one which is a new thing. Somebody discovers a new thing, or somebody promotes a new idea, that is called invention. So invention is not a product. Invention is a virtual thing. It is an imaginary thing. Just you are getting some idea. This can be like this. If we use it like this, it will be helpful, it will be beneficial, useful, and all that. But all our ideas and thoughts virtually, will it have any use in the real world? It will not have any use in the real world. So what will be useful in the real world is a product which you can touch, which you can use it for making your life easy and all that. So what is this difference between now invention and innovation? Invention is an idea, an imaginary thing, a virtual thing. You cannot touch it and you cannot feel it. But innovation is the one how you convert these ideas into a commercially viable, useful product which will be really appreciated by the person who will use it, or a people who will use it. So this is the difference between invention and innovation. You should always be careful while using this word, because both the words are not synonyms to each other, and both the words are not interchangeably. You can use it. So here today we are going to talk about the innovative things. Innovative things means how you can use your innovative plan to convert an inventive things into a product which will be really helpful to the people to solve some problem, to make their life easy, and to commercially viable, which will be required by the market and all that. Okay. So today we have our expert, Dr. Ajay. So Dr. Ajay is not only an expert today, he is a well-wisher for our Amity University as such. So he has a very unconditional support, love, and affection towards all of us. So he is a constant support and inspiration to all the young generation today. So you listen to him, how in very young age he has become a very great person. Now he is working in the Delhi Institute of Pharmacy in a government organization. In a very young age, he was able to bag that position. So that is not only because of the knowledge, because of his attitude and other things. So you should learn from him, not only knowledge and other things also. I thank profusely Dr. Ajay for going to spend his valuable time with us. He is very busy person, but because of the love and affection that he has on our institute, he has consented to deliver this talk today and spend his time with us. I also appreciate Dr. Naveen sir for arranging this program in a very meticulous way. My sincere thanks to Dr. Naveen sir also, who is also our IIC coordinator. Thanks a lot. I sincerely believe you will have take-home points today, which you are going to learn, which you don't know earlier. Thank you very much. All the best.
Thank you, madam. Now I request Dr. Naveen Sharma, coordinator of IIC, to introduce our today guest, Dr. Ajay Sharma.
Thank you, sir. Good morning to all. We are delighted to have the gracious presence of our guest speaker, Dr. Ajay Sharma, of the today's event, organizing under the institution's Innovation Council activities. Dr. Ajay Sharma is currently working as professor and head, Department of Pharmacognosy, School of Pharmaceutical Sciences, Delhi Pharmaceutical Science and Research, University, New Delhi. He has completed his graduation from School of Studies in Pharmaceutical Sciences, Jivaji University, Gwalior, and Master from Dr. Hari Singh Gaur, Vishwavidyalay Sagar. He has completed PhD from Bharati Vidyapeeth University, Pune. He has been awarded postdoctoral research fellowship from Yeonam University, Republic of Korea, senior research fellowship from Department of Biotechnology, New Delhi, and junior research fellowship from UGC, New Delhi, India. Research area includes drug discovery and development from natural sources, medicinal plant biotechnology, bioactivity guided by fractionation and bioconversions. Sir has published more than 80 articles in peer-reviewed journals of national and international repute, and also presented more than 100 papers in national and international conferences. He has contributed 5 books and 10 book chapters and published 5 Indian patents. He has been awarded with research grant from CCRUM and MPCST. He has been awarded with Best Teacher Award, APP Young Achiever Award, Innovation Award for Scientific Research, and Best Research Paper Award. He is a member of various national and international bodies, recognized as top 2% scientist by Stanford University List 2020, nominated as principal member, Unani Sectional Committee, Bureau of Indian Standard, New Delhi, India 23. He is a coordinator of NAC, NPA, IQAC, and UGC Nodal Officer and coordinator of BPharm, Ayurveda and PG Diploma in Plant Tissue Culture course. With this brief introduction, I now request Dr. Ajay Sharma to take over this session. Sir, please.
Thank you, Dr. Naveen, for your kind introduction and words. Of course, this is not needed at this stage because I am not joining you people as an expert, as told by our director, Dr. Mohana Lakshmi. Still, I am the part of MIT University, Gwalior. You can consider me from there. I am sitting here and I am sitting here for the benefits of MIT University, Gwalior. So for that purpose, that much big introduction is not needed. I am not new to this organization, and I have been associated with this organization since long. In the year 2020, no, not 2020, in the year 2019, me along with Dr. Naveen and other faculty members, we have migrated from Shriram College and we have joined MIT University. And that time we were involved in the development and the establishment of this beautiful organization. So once again, I am thankful to Dr. Navi for your kind words. I am very much thankful to the Vice Chancellor of this university, AVSM retired, Mr. V.K. Sharmaji, and the Director, Dr. Mohana Lakshmi Ma'am, who has invited me and given me this opportunity to join this particular guest lecture. Thank you very much, madam, for your kind word as you talked about, big words about me. And you have beautifully covered the concept of innovation and innovation in your introductory remark. I am also highly thankful to the members of organizing committee who is involving in the organization of this particular event, and other teaching and non-teaching staff members. And of course, I am happy to see my all the students who is sitting in this seminar hall of MIT University. Earlier, I was the part of this family. Still, I am here. So I can begin with my presentation. What I am talking about to you people for today's. That is the process of innovation development. As you know, we all are working in the area of pharmacy, which is full of huge opportunity when it comes to invention and innovation. And as I am working in the area of natural product drug discovery, so I try to basically touching upon what are the key factors and principle in the area of drug discovery and development when it comes to natural products. So if you see, I am audible and visible as well. My slide is okay, Dr. Naveen?
Yes, sir. Yes, sir. You're audible and visible. Slide is
Okay,
working also.
Yeah, thank you. So as told by our director, Dr. Mohana, what is invention and innovation? And if you are coming up with the new concept and idea, it means you are inventing something. And when you are converting that particular new concept and idea in the form of some product, some process, that becomes innovation. And that will be basically for the benefits of the mankind. And the innovation and the invention, these are basically the theme or the concept that you can commercialize further. So if you see the research perspective, that is one of the very famous quotation that I took from some of the research article. The research is basically the transformation of money into the knowledge, because you have to invest too many things while doing the research, and innovation is the transformation of knowledge into the money. So basically, the young India, or when we are celebrating, the Azadi Ka Amrit Kal after 75 years, the Indian government, they are basically promoting the innovation and entrepreneurship, particularly with respect to the young mind. So as all of you are, the budding pharmacists, you can invent and innovate the things, and by this way, you can transform and convert your knowledge, and you can earn huge money as well. So this is basically the concept of research and innovation. And when we talk about research or innovation, ultimately one of the objective while doing all these things are basically the business perspective. How you can think how India can become a global hub, particularly during the COVID times. So this particular concept, it is related with the ideas and the solution. You have to come up with the new idea, which should be strong, and you have to think whether I have to move forward with this particular idea or not. And what is the business proposition which is linked with this particular idea, means whether the idea is strong enough to move forward or not. And what are the business feasibility which is linked with this particular idea or innovation, whether business will be feasible or not. And if it is feasible, you can move forward and you can make a concrete business plan to bring your innovation or idea on paper, and then you can go according. So n number of things are basically required while doing all these things and exercise, and of course the understanding of the earlier technology as well as the technology which is prevailing nowadays is must, because for innovation and for research as well as for the invention, the understanding of these technology is very must and important. And from here I will tell you how we have started innovation, not now. This is the global mission. Even in the earlier time, we people or our ancestors, they were involved in invention and innovation very beautifully. I will show you these some slides and you will come to know how the invention has been done, how the innovation has been done. And while sitting or working at MIT University, you can innovate n number of things and you can come up with the product which is the need of us. So what are the various technological development that has been taken place in the area of natural product drug discovery? You see this particular slide, how it looks like. If you see the herbal drug or the natural product based research, how the revolution or the development has been taken place, you can see in this slide, I am talking about the first revolution and the second revolution. See the time of Ramayana when the Lakshmanji was hit by Meghna, and for his healing, how the Hanumanji, he went to basically the Himalayan region and how he has collected basically the Sanjeevani booty, and how the Ved of that particular time, Susain Ved, if I am not wrong, how he cured and gave new life to the Lakshmanji. So this was the concept of invention and innovation. That time, the technological development was not there, but whatever the technology was there, whatever the science was there, the peoples were using that technology and the science for the benefit of the mankind. And at that time, the information is given in various our religious and holy books. So that time we were having various Vedas, like Rig Veda is there, Sam Veda is there, Atharva Veda is there. So that was basically the Vedic canon. So where different scripts were available, the method of preparations of jadi booty or the herbal drugs were available, how they were triturating the things, how they were preparing the juice. But even after that, that time the myths were available as well. But that time there were no regulations existing. There were no Ayush regulation. There were no FSSAI. There were no US FDA. But even after that, the technological development was there. Now, if you will come to the second revolution, which is comparatively, little bit more advanced, you will understand the concept of the cultivation of the medicinal plant from their wild resources. The peoples were, preparing the herbarium. They were doing, the ethnopharmacological evaluation of the drug, though the concept was not, ethnopharmacology, not that much well developed, but peoples were using, the concept of pharmacology depending upon their the traditional claims. And again, that time in the second revolution, the concept of Drug and Cosmetic Act came. Now, the trend of third revolution came, where we were talking about the extraction process. We were talking about the microscopy, where the scientists, they were using different techniques, how to prepare extract and how to basically identify the cellular structure, which is basically very important even nowadays, and how they are going for the fractionation and characterization of the phytoconstituent and high throughput screening of novel leads. And still, the myths were there, because even in the third revolution, the methods of standardization was not well set. And some of the regulation came with respect to dietary health, uh, and Education Act, that is DHSHEA, particularly when we talk about nutraceutical or food supplement. The European Union, they also came up with some of the regulations. The schedule came if we talk about the our country, and in China, the Food and Drug Administration related some of the regulations came. So that was basically the development during third revolution. And if you see the basically just a third revolution, this slide is again linked with, and this all are related with how the herbal drug discovery and development has been taken place in a systematic way, where we can start the innovation from the collection of the drug material. That is again not that much easy. A lot of innovation and efforts are required while selecting the particular herbal drug for therapeutic and curative purpose. So you can start with the collection. Drying methods are there. The teachers are basically teaching you the various methods of drying, but what are the innovative methods of drying, which is the need to save the phytoconstituent, because the several phytoconstituent, they are thermolabile as well. And when talk about the extraction process, we know the succinate is there, the percolation is there, the maceration is there, the homogenization is there. But nowadays, as per the demand, we have to move forward, and we have to innovate some advanced method of extraction who are basically environmental friendly as well, particularly with respect to microwave assisted extraction and ultrasonication. So they are considered as the green technique, which are different from the traditional method of extraction. So in each and every step, innovation is involved. You can do innovation, okay? So you can come up with the new ideas and you can execute it. Whatever the facilities we are having in MIT University, that is also sufficient to innovate something new. And similarly, you can move forward for the separation and the purification of phytoconstituent. Then you will get the single phytoconstituent, identify it, go for the biological evaluation using clinical trials from phase 1 to phase 4, and take the FDA approval or whatever approval is there with respect to the drug material, and you will get the product that will be ready to launch into the market. So the innovation is there, the invention is there, the business potential is there. That's why you have chosen very good topic for discussion during this webinar. And this is the fourth revolution. Again, you see the new concept came in the earlier three revolution. The concept of the script, the Vedas were there, then the microscopy, then something extraction was there. Now in the fourth revolution, which is the very new revolution, the concept of plant biotechnology for micropropagation and for enhancing the yield of secondary metabolites, biotransformation, confocal microscopy, electron microscope, reverse pharmacology, they came. And now the standardization with respect to the herbal drug, that is also came. So we are standardizing the drug material as per various guidelines, particularly the WHO and other guidelines. And if you see the post-COVID era, just 2-3 years when we all were struggling with the COVID-19 pandemic, then the concept of genomics came. When the world, when the mankind, when they were struggling with for their survival, the concept of genomics, the concept of the ancient Indian system came, and they have provided good relief, and they saved the life of so many peoples. And the concept of green biotechnology is again one of the revolution during post COVID-19 era. Similarly, the chloroquine, that is the research from our university. We people were involved doing this. That is the perfect example of innovation what we have in our university. And the drugs from microbial origin, and one of the very recent discovery that happened, our honorable Prime Minister Sir he has inaugurated TKDL Center in Gujarat. So that is basically the very good, you can see the reputation of our country, how the WHO, they have established traditional medicine, traditional knowledge and medicine center in Gujarat under the leadership of our honorable PM. So how the development has been done, you can see here. Now, the concept, the innovative or the novel concept is how the biomarkers, they are related with drug discovery with the use of a reverse translational approach. This is again the innovative approach. The scientists, they are coming forward and they are using these concepts for various prophylactic and therapeutic purpose as well as for diagnostic purpose. You see how the COVID-19 diagnosis or test has been done using the concept of PCR based kits. So they all are basically the biomarker based drug discovery and a reverse translational approach. So what are biomarkers basically? These are the markers which are present in the biological system. Like insulin is one of the biomarker for diabetes, you can think. Clear? So similarly, various proteins are there, various enzymes are there, which are considered as biomarker, and they are used for various purposes. So they may be prognostic marker that may be measured by some of the technologies, and they will tell you how the risk has been associated with the particular disease. Yeah. Like for example, you can see the glycated hemoglobin. If it is more, then you have to be very careful about the setting up of your diet with respect to diabetes. So similarly, the predictive markers are there. By using these, you can think about your treatment strategy or approach, the theranostic markers and the surrogative markers. So all are basically the biomarkers. Similarly, if you want to know the health of your kidney, immediately you have to think about the creatinine level. Clear? So all are basically the considered as very important biomarkers. And how you can use these biomarkers in clinical practice. So as I told you, they are very important for diagnostic, for prophylactic, and for therapeutic point of view. So these are the various examples of biomarkers, okay, various enzymes or proteins that you are looking for. These are the major challenges in the practice of medicine. The concept of preventive medicine that was primitive in the earlier time, prevention is better than cure. That was our assumption. Now, epidemiological studies came. The database related studies are going on, and health for all with the use of data science or gene therapy related research. That is the concept nowadays. This is the evolution of various Indian systems of medicine, as you all are aware about the Ayurveda, the Chinese, the Unani, the homeopathy. They all are there. The modern versus the herbal drugs. The concept of modern and herbal drug, and all these concepts you have seen during the fourth industrial revolution. Different challenges in the drug discovery. When we talk about the herbal drugs, we have to face, or we are facing a number of challenges. When we talk about the standardization, the things are not available on paper. So how you will do that? Though nowadays certain guidelines are there to whom you can begin with. And apart from that, once you talk about related to the pharmacological point of view, if you want to establish the mechanism of any particular herbal drug or their biomarker, you have to take care about their quality, safety, efficacy, along with the toxicological studies or n number of other things, particularly related to PK and PD. So these are the challenges with the herbal drug discovery, and that we have to set. Now the concept of the personalized medicine came, and there you are studying the genetic profiling of the particular individual, that is the diseased individual, and accordingly you are designing the treatment. So if the genetic abnormality is there in the childhood when the baby is coming for birth, that time you can treat that particular individual by by seeing or studying their the genetic profiling, what type of genetic effects are there, that you can fix that time. Or even after the birth, if we are having certain sets of the tests and procedures that you come up with, and you can identify what are the enzymes or the proteins which are defective in the particular individual, and that you can basically rectify it. So that is the concept of personalized medicine. Now you see over here the reverse translational approach. When we just writing the prescription, that time just see the concept or try to use the concept of reverse translational. Earlier, whatever the patients, whatever, whenever they are visiting the doctor, doctor basically used to give the prescription. And even nowadays, the same system or practice is going on. But see over here, if you are going in this way, your prescription will become very effective, and you can handle and treat the disease in a better way. So nowadays, what we are doing, first of all, we are identifying the targets. What are the targets? Suppose if a person is struggling or suffering from diabetes, so first of all, you have to identify the targets or the markers or the genes or the protein which is responsible for the prognosis of the diabetes. Like GLUT4 protein is there, insulin receptors are there, DMPP type of proteins are there. Clear? Different enzymes are there, like amylase is there, kinases are there. They are regulating the glucose metabolism. So with the help of molecular modeling or dynamics approach, we can come to know about the target identification. Then you can go for the validation of the target with the use of some in vitro studies, and then go for the in vivo study, set and establish the protocol, then go for preclinical and then clinical from phase 1 to phase 4. Clear? And again, you can go for the personalized medicine. Go and see the genetic profiling. Go for the diagnostic development. What is going nowadays, particularly NGS, that is next genome sequencing, where you can come to know about the gene profiling of the particular individual, and accordingly you can design or set your prescription. Now you can see this particular slide, how the drug designing process has been taken place, where you can start with the gene chip experiment, go for the identification of proteins, then project selection decision, what type of project is the demand nowadays, then go for the optimization of protocols, whatever they are there, then go for HTS, that is high throughput screening, because n number of phytoconstituents are there, you can't go individually with all of them. So using this particular technology, at a time you can screen all the phytoconstituent, and depending upon the result, you can go with the best one. And of course, the combinatorial experiment, the structure activity relationship, pharmacophore modeling, ADME studies are there, by which you can move forward and you can contribute in drug discovery process. You see the drug discovery and the development. So in the 20th century, first we did pharmacology and then we move for genomics. But now the trend is just reverse. First, you have to understand the genomics of the particular thing and then do the pharmacology. So target identification is must, where you can go with the bioinformatics, then the receptor-based approach, then the ligand, then the screening of the compound, find out the lead compound, go for the bioactivity guided fractionation or the biological activity, then again go for the screening of those compounds, and you may get the drug candidate. So this is the strategy which we are going with. Now you can see the classical herbal drug research with respect to the reverse pharmacological approach. So whatever the claims are given in our books, we are reading that, we are come up with the information. Accordingly, we are preparing fractions. Then we are going with bioactivity guided fractionation. Then we are coming up with the standardized extract. And then once you are having that standardized extract, you can go for the clinical trials. So that is the thing which is going with the classical herbal drug research. Each and every step, what we are discussing over here is full of innovation. You can invent and you can innovate and you can come up with the product. So each and every step is related with the concept what we are discussing today. You see the concept of globalization. So think global and act local. That's why we are using, or we believe in, our ancient Indian system of medicine. We are worrying about the health of the globe. So now the Indian country, our country, we took the lead and we are basically taking care of the world health, and we are promoting our ancient system of medicine, and TKDL inauguration is one of the example inaugurated by WHO. So I will tell you here some of the clinical study based example where E-letrolale is one of the drug, that is effective in glycemia control. Yeah. So this is the reverse pharmacology approach before the COVID and during the COVID era, where we have, used the concept of bioinformatics and molecular modeling, and that result with the lead candidate or the drug candidate. You can see how the sugar label has been maintained by the herbal drugs. You can see how the glycemia is controlled in various groups. Now this is again the DNA migration or DNA damage related assay, which is the COMET assay. Using this, you can come to know whether the DNA damage is there or not once the person is basically suffering from diabetes, and if DNA damage is there, how you can reverse it. So all these things that you can read or understand with the concept of COMET assay. You can see this is the effect of one of the phytoconstituents, Yersinia marin, on the lipid profile of experimental rat. So this particular glycoside, it is not only the taking care of your lipid profile, also it is regulating blood sugar level. You see their mechanism of action. This is the need of our. Now we should not talk about like Momoedica carencia is anti-diabetic in nature. No. Now we want what is the mechanism of Momoedica carencia in diabetes. Okay. Now the 2 decades of RAS, that is renin angiotensinogen system, how the drug discovery and development has been taken place with respect to this particular enzyme, especially during the COVID time. Whatever the post-COVID complications we have seen, difficulty in breathing, fibrosis of the lung, the main cause is inflammation, and this is one of the enzymes which is involved in all these pathophysiological changes during COVID-19. So this is the structure of virus. I am not going in detail. It is basically containing Skypes along with nucleocapsid. This is the ACE2 receptor. So how the ACE2 enzyme, how it is binding with the particular receptor. So this is the virus and the protein or enzyme interaction. So how the development or the interaction has been taken place. This is linked with the innovation or how we come up with the product during the COVID time. That's why I am discussing all these things with you. You can see over here how we came up with the product during the COVID time. So first we have understand the structure of the virus, how it looks like. Then we went for the role of ACE2 in COVID. So we went for the literature survey and number of articles were available, and after that we come to know we come to know what are the probable phytoconstituent which are having ACE2 inhibitory activity, because that is the enzyme which is linked with the pathophysiological changes. So we went for literature survey. We find out the plant material. Clear? We come up with the phytoconstituent, and we have selected 195 phytoconstituent. We went for molecular docking and dynamics, and we have found 26 hits out of 195 phytoconstituents. So these phytoconstituent again, they went for visualization and scrutinization, how the ligand has been selected. So you have to dock your phytoconstituent with the target what you have find using various techniques. So the drug and the receptor binding, that particular binding, that particular dynamics you have to read using the concept of molecular docking and the dynamics. And 26 hits we have selected, and we have screened those particular hits further. So again, we can narrow down these 26 hits, and again we can narrow down these 26 hits, and we can end up our experiment with only 8 to 9 hits. And those particular 8 to 9 hits, we have utilized for the product development. So, in these, hits, what I am talking about, rutin is one of the, phytoconstituent that showed better docking score above 10. And, with respect to rutin, we have also compared synthetic drug which is active against, or which is considered as ACE2 inhibitor. So the rutin, that is from the herbal origin, and one is basically the drug from synthetic origin. We just dock this particular drug to the particular enzyme, and we came to know that rutin shows better binding affinity, and you can see the docking score of rutin as well. Now remdesivir also, that time during the COVID time, that was in the peoples were using remdesivir without knowing anything. We have compared the docking score of our phytoconstituent with the remdesivir as well, and you can see the remdesivir, the docking score is -6.7, which is quite less than the docking score of hesperidin, which is another phytoconstituent. It means it is having more binding affinity with respect to the available enzyme, proteins, and amino acid, which is the need, especially in case of COVID discovery. Now, these are the various plant materials that we have selected for our product development. Solanum nigrum, this is one of the drug. Acalypha indica. So see the phytoconstituent, what they are containing. So they contain flavonoids, they contain phenols. So depending upon the literature, earlier we have selected all these material as such, but we have narrowed down all the phytoconstituent which are available in all these 8 or 10 plant, and finally we have selected Solanum nigrum and Acalypha indica along with their phytoconstituent depending upon their docking score. And that was basically the study conducted. This is basically the drug chloroquine zinc that our university, under the leadership of our Honorable Vice Chancellor, we have come up with this particular product, and this shows their effectiveness, especially during post-COVID complication, because it has enormous ability to arrest the ACE2 enzyme activity. So it shows ACE2 inhibitor. You can see over here, various studies has been conducted. What is the effect of chloroquine zinc, which is our developed formulation, on viral entry? And if you see with the concentration, it decreased viral entry to the particular individual as well. You can see this particular graph. The IC50 value has been also calculated. You can see how the chloroquine zinc, how they reduce viral load in Syrian hamster model. That is again, we have checked, and the activity was good even after the 4th day. The viral load decreases significantly. You can see the after the administration of our formulation, how the viral load has been decreased in another patient. You can see it over here. The results were absolutely significant. You can see this is again the study what we have conducted, because during COVID time, the patients, they were suffered a lot with respect to breathing related issues. So with respect to MMRC, that is again one of the biomarkers which is related with dyspnea, means difficult breathing scale. So again we have analyzed the activity of our drug with related to or with respect to standard drug. So you can see over here from the activity was maximum from 7th day to 14th day. The level or the dyspnea level, it went down significantly. And now you can see we lost the taste as well as smell during COVID time, especially the patient who were struggling with all these things. And the taste and smell, it came again when the patient was administered with chloroquine. So they were recovered from anosmia and ageusia, that is the loss of smell and taste. You can see the effect of chloroquine formulation on various inflammatory biomarkers. So we have determined the effect of our drug on various inflammatory markers like interleukin, C-reactive protein, LDH level, hemoglobin, and a D dimer. That was again one of the biomarker during COVID time. It is very useful for diagnosis. D dimer, that was the prognosis marker. So this was the strategy what we have adopted with respect to our herbal drug, which we have selected for chloroquine zinc development. COVID-19 herbal drug approach, we have targeted, or we have selected ACE2 as a main culprit. And with respect to ACE2 enzyme, we have selected phytoconstituent on the basis of, their score. We went for herbal formulation, and again, we went for various, antiviral and clinical trial based assay, and we come up with the new formulation which is available in the market. Now, our university is in the process of getting regulations from the Ayush Ministry. The drug is available in the market in USA as well as in European and UK, but in India, before going to the market, we need some approval. So that is under process. So this is all about my presentation. So once again, I am very thankful to the director of AIP, AUMP, and the coordinator of Institute Innovation Council, Dr. Naveen Sharma, and the faculty members and the students for patient listening to me. Thank you. If you are having any query, please ask.
Thank you, sir. Thank you, sir. It was wonderful knowing your views and opinions on this topic, the progress of innovation, development, and technology readiness level. So from the audience, any doubts, please? Yes, sir. Yes, sir.
Hello.
Hello.
Yes, please ask.
Thank you so much, sir, for this wonderful talk. My name is Narendra Kumar. I'm an assistant professor in pharmacognosy department. So thank you very much for this wonderful talk. Sir, I have a small question regarding the chloroquine. You said that chloroquine targets the ACE2 entry entry. So how do you know that it selectively target the COVID or SARS-CoV-2 ACE, not the human lung? Because the implications of the other protein like PL3 Pro, M Pro, RDRB Pro is much more than the AS2 in COVID-19. So how do you know that is selectively targeting the ACE2 inhibitor of COVID?
Yes, actually the study with respect to that protein, other proteins that you are telling or mentioning, that is also in process. But depending upon the, the structure of the virus, and depending upon, whatever literature were available, because COVID came first time, no one was aware about the COVID and its implications and what is the pathophysiology of the disease. So whatever the research paper came, whatever the articles were available that time, we went through all. And we also read the pathophysiology of ACE2, particularly with respect to post-COVID complications. So the post-COVID complications or the ACE2, if you see their role and pathophysiology, they more or less they are related with the inflammation, and because of that inflammation, one of the complication is pulmonary fibrosis as well. We went for the literature and we come up with the conclusion that it plays a very important role in inflammation as well as in pulmonary fibrosis. Though other proteins are again there, but the attachment of virus with respect to this particular protein or enzyme is one of the key factor for viral entry. Though other proteins are there, but we have selected this particular protein because of n number of reasons. The first reason is it is very important for viral entry. Second important thing is play a very important role in inflammation and in pulmonary fibrosis. And that we have proven by using some experiment as well, how beautifully it control the level of various inflammatory mediators, and how we can reverse or reverse the the level of D dimer and other things as well. So the n number of experiments we have conducted, and initially we have launched this particular product as a nutraceuticals because we don't have any regulatory approval from the Ayush Ministry, though we are in the process. Once we will get the approval, then we are trying to launch this particular product as a drug, phytopharmaceutical, that the concept which is prevailing nowadays. So that is basically the case. So there are so many reasons important for viral entry, important for inflammation, and important for the pulmonary fibrosis that was one of the major cause of deaths during COVID-19.
Thank you, sir.
Yes.
I have another question. We are talking about innovations. So the changing technology, artificial intelligence, machine learning, how do you think that they can aid in the natural products drug discovery?
Definitely, this is the perfect example of molecular modeling, what we have discussed. So before going further, you can you go for the literature survey, find out the phytoconstituent, draw their structure, and accordingly find out the possible targets, what it can or where it can bind. So this is the perfect example of the computational based drug designing. And definitely it is helping a lot. That is the concept of reverse pharmacology. Initially or earlier, what we were doing, we were making the extract and we are testing those particular extract using various in vivo model. But now it is not the case. Why unnecessarily you are extracting the drug material, and why unnecessarily we are basically torturing the animal? It's better first you find out the targets where your phytoconstituent can bind. So for example, if I am looking for DPP4 inhibitor, which is one of the very important target for maintaining the blood glucose level in diabetes. And if we know from the literature, if we know what are the possible phytoconstituents which can beautifully bind a DPP4 inhibitor, if we select those particular phytoconstituents, and by keeping all these things in our mind, we can go for extraction, then it would be better rather than going for, closing our eyes and going for the extraction what we did earlier. So better first find out the targets and find out the phytoconstituent which can beautifully bind to your target, and then move further. And that's why the concept of the personalized medications came. If you are having fever, if you are having diabetes, if you are having hypertension, Alzheimer, whatever the problems you are having, the doctor, they are prescribing the medications. Why to do that? Why not to first understanding the concept of the personalized medicine and then move further. So first of all, the things are going in that way. Just read what is the genetic problem or genetic profiling of that particular individual, which gene is defective. Accordingly, you design the medicine. That would be far better.
So, so any more from the students to interact with the
Any
guests?
questions from the student side?
If you have more questions, you can post it as well. I will reply whenever possible.
Sir, maximum drug made on the basis of the treatment of body cells. What about the treatment of the virus disease? Because viruses are made up of without cells.
Yes, but they are active once they will enter in the biological system. That time they become active. So whatever drug, especially in the COVID time, that is RNA virus, COVID-19, we have designed drug accordingly. So, but before that, you have to understand the structure and the nature of the virus, and then you can come up with the drug. Outside it is, you can consider it as a particle or non-living thing, but once it will enter, then it will show their real colors. And accordingly, you.
Okay, sir. So thank you, sir. Thank you very much. So now, sir, we would like to give the virtual felicitation to you, sir, for sharing your views and expertise in this session, sir. So thank you. Now I would like to call Dr. Rajesh Sharma, sir, to propose the vote of thanks for this event.
Thank you, sir, for giving me this opportunity for vote of thanks. A very good afternoon, one and all that are present here. Firstly, I would like to thank our guest speaker, Dr. Ajay Sharma, for your expert views and opinion for the progress of innovation, development, technology for the herbal drug, nanomedicine, and drug discovery. I am also thankful to all the participants for joining this session. I am sure that all the participants must have benefited by attending this lecture and these events. So I would like my sincere gratitude to our Honorable Vice Chancellor, Lieutenant General V.K. Sharma, sir, for permitting us to organize these events. I am also express my heartfelt thanks to our Pro-Vice Chancellor, Dr. M.P. Kaushik, sir, for his profound guidance. I extend my sincere thanks to unwearying and unskilled support receiving from Director of Amity Institute of Pharmacy, Dr. S. Monalakshmi ma'am, making this event successful. And I also thankful to all the faculty of AIP, IIC coordinator, HOD of various departments for their constant support and guidance. I am also thankful to IT department, administration, and non-teaching supporting staff of Amity University for providing us with all the possible support and guidance towards the organizing this event. Once again, on behalf of Amity Institute of Pharmacy, thank you all for making this joint this session. Wish you all the pleasant day. Thank you all.